What I'm going to try to do today is I'm still going to cover the basis like I did last time, but I'm going to tell you a lot of things that happened between last December and now, where we stand with vaccine rollout, which vaccines we actually now have, and what we know about them in terms of efficacy and safety. And also I will talk a little bit about these variants that we hear, and I'll show you some data about the New York variant. I guess you're interested in that. Coronaviruses are a family of RNA viruses. They're called coronaviruses because they have a crown-like appearance in an electron microscope, and you see that here on the right side. So you have these balls and then these protrusions, which appears like a crown. They are relatively large in terms of their genome size. They have about 30,000 base pairs or letters. And this is shown here on the right side. So we have the genome on the inside of the particle, and this genome is covered by a protein that protects it, nucleoprotein, because RNA is pretty unstable. Then we have a lipid envelope on the outside. We have two more proteins in that envelope, and then we have the spike protein on the surface. And this thing is about 120 nanometers in diameter. And in comparison, a human hair is about 80,000 to 100,000 nanometers in diameter. So they are pretty small. We have 4 human coronaviruses that are causing common colds. We know of them for a long time. They're not particularly dangerous. And we have known about 2 zoonotic coronaviruses. So zoonotic means that they come from an animal and infect a human. And those are SARS coronavirus 1, which was caused an outbreak mostly in 2003 and infected about 8,000 people and killed 800 of them. And then we have the Middle Eastern Respiratory Syndrome coronavirus, or MERS, which still jumps from camels in the Middle East into humans, but there are not that many cases, and it doesn't transmit very well from human to human. Those 2 are dangerous. They have case fatality rates of about 6%, sometimes higher. And so then the new virus came around and was quickly identified on January 10th. Chinese scientists at the Fudan University who had sequenced the virus actually posted the genome online, the sequence. And if you have these sequences, you can build phylogenetic trees, which are basically very similar to basically genealogical trees. And so it's easy to figure out what the relatives of this new virus was easy to figure out what these relatives of the new virus were. And you can see here the red star is the new virus, and the yellow star here is our old SARS coronavirus 1 from 2003. And so because there was this relationship and the disease was similar, the virus was termed severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2. And the disease was termed coronavirus disease 2019 because it showed up already in 2019. And what is important to mention here, and we'll get into that, and it's super important for vaccines and immunity, is this spike protein on the surface, these protrusions that make it look like a crown. And so this spike protein is used by the virus to dock onto our cells. Specifically, here is a crystal structure of this spike protein, specifically this red part here, which is called the receptor binding domain. So this interacts directly with ACE2, which is a protein that our cells express. And then the virus can actually bind to that and enter the cells. Of course, if an antibody, which is, as you all know, a protein molecule that our B cells make, binds here, and the virus can't bind to our cells anymore, and the virus can be neutralized. We knew that already from other coronaviruses. And so just to show this, when we inhale some of this virus, it lands on our mucosal surfaces, and then it finds itself a cell, and it just docks onto the surface of the cell. It goes inside, and it starts to basically take over the cell as a large replication machinery, and it forces the cell to make more virus. And of course, at some point, the cell dies, the virus is released, and there's many cells in our body, and so the viruses just go on to infect other cells, and you have more and more virus in your body, and that's basically what makes you sick. In some cases, unfortunately, that leads to severe disease and death. The fortunate thing with this virus is that the majority of people who get sick recover from the virus. And if you recover, you also develop an immune response against the virus. And this can be based on T cells and B cells. So T cells are cells that kill infected cells and therefore prevent the virus from further spread. And B cells are cells that make these antibodies, these little proteins that can neutralize the virus. This is, of course, a very simplified version of how this happens. But if you go through a SARS coronavirus 2 infection, your immune system will respond and you make these T cell responses and antibody responses. And so this is how it typically looks like with antibodies. So on the x-axis here, you have days since infection. And on the y-axis, you have basically the magnitude of these analytes. Basically, initially, you get infected and the virus starts replicating in your body. There's more and more viral antigens or viral proteins, more and more RNA. This is what we detect by PCR. And then if you're doing well, your body is starting to clear the virus at the same point and actually using your immune system. And you see the immune system and the immune response coming up here, and the antibody levels go up and then they stay up. And so these antibodies don't go away after infection, they stick around and they can protect you from getting infected again. And so now if you have the situation where you already have these antibodies and you get exposed to the virus again, the virus lands and basically lands on your mucosal surfaces, but there's already these antibodies that now bind to the spike protein and cover it, and so the virus cannot attach to your cells anymore because it can't bind anymore. And so the virus gets neutralized and you're protected. And just to sum up what we know by now, and we know a lot about the immune response to this virus by now, we see that antibodies target all kinds of proteins in the virus, specifically also the nucleoprotein and the spike protein. We know that the antibodies that target the spike protein can neutralize the virus and that can protect you. And we already know that these antibodies against the spike protein correlate with protection, meaning that if you got an infection and you have these antibodies, your risk to get reinfected is very, very small. It can happen, but it's very rare and you're very well protected. We also know that we get T cell responses. There are different types, CD4 and CD8 T cells, and they develop also after infection. But of course, you don't want to go through an infection to be protected from the virus, right? And there's other ways to do that. And of course, that's vaccination. And very early last year, people started to develop vaccines against SARS coronavirus 2. Actually, the day the sequence was released, Kizzmekia Corbett at the NIH already designed the vaccine that would become the Moderna vaccine. So she did that in one day, and that vaccine went into clinical trials on March 16th and was then licensed in December. So what type of vaccines do we have? Right now, these 4 types of vaccines are the vaccines that are used in most parts of the world. We have inactivated vaccines. So these are very classical vaccines where you take a patient isolate, you grow it in cells in a laboratory, you inactivate the virus, usually with chemicals, you purify it, and then you use that as vaccine. And you inject that into your muscle, and then your immune cells find that virus, which can't do any damage, but then you make an immune response. It's a very old-fashioned way of making vaccines. Hep A vaccines and flu vaccines are made like that. These vaccines are currently not used in the US, but they are used in most parts of Latin America, in many Asian countries, also in African countries, and they are actually also working relatively well. And then we have recombinant protein vaccines, which are also relatively classic way of making vaccines. So you just take the sequence of the spike protein, and in the lab you have cells that can now produce this spike protein, but are not making the virus. You can purify this, you inject that, and that's your vaccine. This is done for hepatitis B viruses, for human papillomavirus vaccines, but also for one type of flu vaccine that we use in the US. And then we have the newer technologies, viral vector vaccines and mRNA vaccines. And I'm going to describe a little bit better how they work, because people are curious about that and sometimes worried about these new technologies, while with the classic ones, there are not many questions. So let's start with viral vector vaccines. So typically, we use harmless viruses for this. In the case of SARS-CoV-2, it's mostly adenoviruses, which are viruses that cause common colds and sometimes diarrhea. Basically, you take away parts of the genome of this virus, so it can't even replicate anymore, and you put the information for the SARS coronavirus 2 spike protein in there. And then the virus is used as a vaccine, and so it basically docks onto your cells, goes into your cells, and provides the information for making the spike protein. The virus itself doesn't replicate, but then your cells make the spike and you make an immune response. And those are vaccines that are currently already authorized. The J&J vaccine is based on an Ad26 vector, and we have in phase 3. You've probably heard in the media a lot about AstraZeneca recently. We have the AstraZeneca vaccine here in the US in phase 3, and in Europe that's already widely used. And that's also based on adenovirus vector. In addition to that, the US Army is using adenovirus vaccines since 1971, I believe, in fresh recruits to avoid adenovirus outbreaks that make a lot of people sick, and then they can't complete their basic training anymore. And there's also an Ebola vaccine based on Ad26 that is used in the European Union. How does this work? Again, you get injected with this virus. It binds to your cells, goes into the cell, and doesn't replicate itself, but provides the cell with information to make this spike protein from SARS-CoV-2. And then this is recognized by immune cells, and you make an immune response against the spike protein. The RNA vaccines actually work mechanistically in a very similar way. Here we manufacture an mRNA or messenger RNA, and I'll explain in a second what it is, that codes for the spike protein of SARS coronavirus 2. This is made in a biochemically, it's purified and then packaged into these lipid nanoparticles, which are basically just small fat particles. They're then injected, your cells take this up, and your cellular machinery recognizes the spike protein, the RNA, and makes spike protein. And right now we have two that are currently used in the US, Moderna and Pfizer, and a lot of you probably got one of those two vaccines already. So just to explain a little bit what this messenger RNA is, we have here a cell, and in the cell you have the cell plasma and the cell nucleus. And in the nucleus, we have our DNA and all the information that's needed all the building plans for proteins, right? And the little machines that build proteins, the ribosomes, are outside in the cell plasma. Now, there's a double membrane around the cell nucleus, and the DNA can actually get out of the nucleus. So how does that information for the proteins get to the ribosomes? Basically, the DNA shown here in red is transcribed by a polymerase, by just a small little enzyme, and RNA is made. And RNA is basically smaller pieces of genetic information. It's a little bit different. The DNA is double-stranded, the RNA is single-stranded, and one of the letters is different. But this is then transported out into the cell plasma and is recognized by ribosomes, which are these little machines that make protein, and protein is made. And of course, our cells are full with that, because if we wouldn't make proteins, we wouldn't be able to survive. So our cells are packed with mRNA all the time. And as I explained, for these mRNA vaccines, we just make the mRNA biochemically, package it in these little nanoparticles, and then they are taken up by the cells. The cell machinery, the ribosomes, recognize the mRNA, start to make protein, the SARS coronavirus 2 surface protein. This is then displayed on the cell. And again, the immune system comes, recognizes this, and we make an immune response. And these RNA vaccines are relatively recent development. They have been in clinical development, meaning there are clinical trials with them since 2013 for many different infectious diseases. And one for cytomegalovirus is now moving forward into phase 2. So this is getting developed further. Moderna started to do that in early 2020, and also one of the rabies vaccines is now moving into phase 2. So they look pretty successful, but usually vaccine development takes a little bit longer. There's, of course, a lot of safety data already with these vaccines. Okay, so we have our vaccines. How do we figure out if they work? For that, you do a clinical study, and to measure how well they work, you do a phase 3 study, which is usually a large study that's done in thousands of people, sometimes in a few thousand, and sometimes like for SARS-CoV-2 in 30 or 40,000 people. Typically, you have two groups of people in these phase 3 trials. The vaccine group, those are people that actually get the vaccine, and the placebo group. This is a group of people that usually gets injected with saline solution. Now, of course, you don't tell those people what they are getting injected with. The important part here is also that these clinical trials are usually conducted by independent medical centers. So Mount Sinai, for example, ran parts of the Pfizer trial. Now we're running part of another vaccine trial. We also contributed to the Johnson & Johnson trial, but they're not run by the companies themselves that make these vaccines. They're run by medical centers. And this is usually geographically distributed, not just in the US, but also across the globe. And then there's another important element, which is an independent committee that watches the data. So, you know, the data first goes to that committee, and they make decisions based on that, when to do an analysis, when to stop a trial because there are safety issues. So they are basically independent of that company. And then before you start the trial, you have to actually set the analysis time points, and you have to define what success is. So you can't run the study and then it doesn't look that great, and then you say, it's good enough. You have to actually beforehand say what is good, what is good enough. And then you vaccinate your individuals, and then you follow them. And then you look who is developing COVID-19. And then basically, once there are enough cases, you count and you look at how many cases did happen in the placebo group and how many in the vaccine group. And of course, you want to see that the vaccine group is protected and the placebo group had a lot of cases. And so I'm going to show you the results from the Pfizer trial and from the J&J trial. For Pfizer, there were more than 40,000 people in that study, and they had 170 COVID-19 cases. 162 were in the placebo group and 8 in the vaccine group. That already tells you the vaccine is working because the group were roughly the same size. And they calculated that they had 95% efficacy against symptomatic disease, which means one symptom plus a positive PCR. So the virus was there and the person was sick. And in the 65 to 85-year-old group, which is important because those are people who are at high risk, the efficacy was 94%, so basically identical. And they didn't see any significant safety concerns. And this vaccine is now widely used. And as I said, many of you probably got that vaccine as well. I'm just going to show you how this looks graphically when a vaccine works very well, and 94% is really as good as it gets. Here we have on the x-axis the days after the first vaccination dose, and on the y-axis we have cases, basically accumulating cases. And the vaccine was given on day 0 and then again on day 21. And this red line here is basically people who were part of the placebo group, and you see there's a constant increase in cases. And in blue, we have people who got vaccinated. And you see that around day 10, when your immune system starts to really act up and kick in, we see the separation of the blue line and the red line. And even before the second vaccination, people start to be protected, and this gets more solid afterward. So this is really a very nice separation. This is what you want to see. For J&J, that's a little bit different because J&J, as you know, is just given once. It's the only SARS-CoV-2 vaccine that's given once right now. Here we had also more than 40,000 individuals in the trial. That trial was done in the US, in South America, and in Latin America. In the US, the efficacy was 72%, and here it was moderate to severe COVID-19. So what they had as definition was 2 symptoms plus PCR positive, plus a positive PCR. And so that's actually not that much difference to what Pfizer had, because if you have one symptom or 2 symptoms, and, symptoms are, a sore throat, for example, or having a slightly elevated temperature. They saw 85% efficacy across all studies against severe disease and 100% protection against hospitalization and death. And this is really, really good. Also, no significant safety concerns, and this vaccine is now authorized in the European Union and in the US. So, for example, at Mount Sinai, we're using it right now. Again, just to come back to the safety question, because that often comes up with these adenovirus, Ad26-based Ebola vaccine was licensed the regular way in the European Union already, and these Ad4 and Ad7 vaccines are used in the US military since 1971. They are not even replication deficient viruses, so what the US Army is doing, they grow these viruses, the regular viruses, and instead of just giving them as injection, they give the live virus as capsule, and so the viruses replicate in the gut, you get an immune response, but you don't get sick. And so they have been using that relatively successfully for a long time. Just to sum up what we have now in terms of vaccines, we have the Moderna and the Pfizer vaccine, which are both mRNA vaccines. They're both authorized and used in the US, very high efficacy. We have the AstraZeneca vaccine, which has variable efficacy depending on the study. And you've heard the controversy in the last days about that. And this is used in the UK and the European Union and in other countries. We have J&J, which is the only vaccine here that is only given once, has a 72% efficacy. And again, we are just talking about mild disease here, basically. It's also a viral vector and is used in the US. The next one to come is probably Novotex, which is a recombinant protein vaccine with a very high efficacy. And then in red, we have vaccines that are not used right now in the US. One is Sputnik 5 from Gamaleya Institute in Russia, seems to have a high efficacy. This is also viral vectors. Then we have a number of inactivated vaccines, Sinovac from Sinovac and Sinopharm from China, but also from Bharat in India. And they have 50 to 90% efficacy depending on the vaccine and the study. And then there's also a vectored vaccine from China that's already used in China, but we don't know really the efficacy of that vaccine. So this is what's kind of in late stage or already used. And to go back to these neutralizing antibodies, it has now been shown that there is really a correlation about how much antibody is induced in the subjects and how well the vaccine works. So you can see here the mRNA vaccines are up here, and then one of the inactivated vaccines is down here. And you see it's inducing less antibodies, so it's also not that protective. And the more antibodies are induced, the better the vaccines seem to work. I also wanted to talk a little bit about safety, and the first thing I wanted to talk about is what we call reactogenicity, and these are just basically responses that you have immediately after you get vaccinated, and some of you might have experienced that. Reactogenicity is usually something that's unpleasant but not dangerous. A lot of people have it with other vaccines too, with flu vaccines, all kinds of vaccines that you might get, but we see it's a little bit elevated with the mRNA and the adenovirus vaccines. And this is basically characterized by injection site pain, headache, fatigue, elevated temperature, muscle aches, and general mild flu-like symptoms. And these things are usually transient. They go away after 24 to 48 hours. And again, we see that a lot with the adenovirus vaccines and the mRNA vaccines. And here on the right is just an example. This is from Moderna, from the mRNA vaccine. On the left side here are the side effects at different doses after the first shot. And the interesting part here is that for the mRNA vaccines, the second shot gives you more side effects. While for the adeno vaccines, it's different. The first shot gives you more and the second less. But again, this is not dangerous. It's very transient, but people need to know about it. And then in the clinical trials, all of these vaccine candidates looked good. I mean, they were tested in 30, 40,000 people. But if you have side effects that are more rare, that you don't detect, right? So you detect those after the rollout. The good thing is they are so rare that for the individual, the risk to get one of those is extremely low. But of course, this is all monitored, and so what was found was that when you get the mRNA vaccines, there can be severe allergic reactions. Usually, they happen in people who know that they're already allergic to other vaccines, and the rate that was determined here is that you have that for the Pfizer vaccine, where there are approximately 11 severe allergic reactions per 1 million vaccinated individuals. And for Moderna, it's lower, it's 2.5 per 1 million vaccinated individuals. Another side effect was recently reported. This one is actually not dangerous at all, but it's noteworthy and people freak out when they see it. And this is basically what they call delayed large local reactions. It's basically a rash that you get. The interesting thing is you don't get it right after the shot. You get it approximately 8 days after getting vaccinated. It doesn't necessarily hurt. It's not dangerous at all. It's probably a T-cell hypersensitivity, and it resolves by itself. It just goes away. But of course, people saw that and were very alarmed. And we only see that with the Moderna, but not with the Pfizer vaccine. And the last side effects that were seen or were discussed, because we still don't know if they even connected with the vaccine, are the side effects that were seen with AstraZeneca and thrombosis. So when the AstraZeneca vaccine was rolled out in Europe, they found 7 cases of what they call disseminated intravascular coagulation in 20 million vaccinated individuals, and 18 cases of cerebral venous sinus thrombosis, or CVST, in 20 million vaccinated individuals. And those rates are a little bit higher than what you would usually see, and so they started an investigation. The European counterpart to the FDA, the EMA, looked into that and found that for regular thrombosis, there is no increase. There might be an increase for this CVST, and this needs to be monitored, but because this is extremely rare and the rate wasn't increased that much, and again, it's a rate of 18 cases per 20 million, and some of them, are the same rate as baseline, they recommended the vaccine should still be used because the benefits by far outweigh the risk. And their judgment was that the vaccine is safe and effective. And so far, we don't actually know if there is a connection or not. But as a precaution, and that also tells you that this monitoring is working, they for a few days stopped using the vaccine to investigate. By the way, in the AstraZeneca trial in the US, none of this was found. The other question that very often comes up is, do these vaccines protect from getting sick or also from asymptomatic infections? And there's now more and more data accumulating that actually says, yeah, they're also protecting from asymptomatic infection. Probably not as good as they protect you from getting sick, but they do. And so there was already some data for the Moderna trial where they saw a 60% reduction of asymptomatic infections after the first shot. And there's now more and more data from Israel, for example, that also points in the same direction. We also knew from the animal experiments that were done in non-human primates that vaccinated monkeys could still be infected, but they had much less virus and for a very short period of time compared to unvaccinated monkeys. So right now, the idea is that if somebody is vaccinated, that person can still get infected in some cases, but even if that person gets infected, it's less likely that the person is transmitting the virus to somebody else because there's less virus and for a shorter period of time. But it's possible. And this is actually similar to other vaccines like the flu vaccine or the pertussis vaccine, which protect you from getting sick, but don't necessarily protect you from getting infected. All right, how long does protection last? The honest answer is we don't know yet. The antibody responses look pretty stable. There's some data over 4 months here from Moderna. Of course, 4 months don't tell you much. But this looks like a normal immune response, and we know from other vaccines that protection lasts for years, sometimes lifelong. But even if it would go down after a few years, you can get a booster shot, and we see that we do that for other vaccines too. That's not a big problem. But right now, the assumption is that we're going to be protected for years. If these variants don't throw us a curveball, but I'll talk about that in a few minutes. The other point is a lot of people already were infected with SARS-CoV-2 and they recovered. And the question is, should they now get vaccinated? And if yes, how often? And so we recently did a study on that, was just published in New England Journal of Medicine. And what we found was that people who already had SARS-CoV-2 and then got one shot of the RNA vaccines had a tremendous immune response. This is shown here in yellow. The yellow balls are people who had already had SARS-CoV-2 and then they get their first shot, and within basically 5 to 8 days they make this wonderful huge antibody response that stays high. The second shot doesn't actually help them much, and they have better immune responses than people who had no SARS-CoV-2 infection and then got the SARS-CoV-2 vaccine twice. And we already know that people who had an infection and then got the vaccine also have very good immune responses against all these variants. The downside here is that typically if you had SARS-CoV-2 and then you get the RNA vaccines, you have more side effects after vaccination. Again, those are none of the dangerous side effects. Those are just the side effects that might be unpleasant, but of course, a lot of people who had SARS-CoV-2 and then feel like they're getting sick after the vaccination are reminded of their SARS-CoV-2 infection, and that's not a nice feeling. Okay, and then I also wanted to talk a little bit about these variants, and you have heard a lot about them from the media. The research really started to look into this in December. When they showed up in the UK and in South Africa and then later also in Brazil. What do we know right now? We have 3 that we call variants of concern, and then there's other variants that we call variants of interest. So we're not really worried about them, but we're keeping an eye on them. We have one that's called B117, that's the British origin variant. Don't call it the British variant. We stopped calling viruses after places where they occurred. That's not really good, but it was detected first in the UK. The correct name is B117. That has a change in its receptor binding domain, that little thing that binds to a receptor, which is actually shown here. So ACE2 is our receptor, and this here is the receptor binding domain of the spike. And here is this mutation, N501Y, got nicknamed Nelly. And so what this mutation probably does, it makes helps the virus to bind better to our receptor, and that makes the virus a little bit more infectious. And now it turns out that it's already also a little bit more lethal in some cases. There's no reason to freak out about it. There's an increase in case fatality rates, but it's not like it's not becoming tenfold more deadly. It's a percentage. And then we have B1351, which was first detected in South Africa. It has the same change in position 501. It has this Nelly mutation. It also has 2 other mutations, and specifically this E484K mutation, which is here, which was nicknamed Eric, is a little bit of a problem because antibodies that usually bind here are then not binding anymore, and you lose a little bit neutralization activity. And then we have P1, or the Brazilian origin variant, and that has the same Nelly mutation and the same Eric mutation. but a different mutation at position 417. And it's probably very similar to B1351. We just don't know that much yet about the Brazilian variant. And here's just the mutations again that we see. So the important one for B117 is this Nelly mutation. And then for B1351 and P1, we have three that really matter. Two of them are actually the same and one is different, but at the same position. Where do these variants come from and why do they occur? So first of all, typically, if you deal with RNA viruses, these viruses don't have a proofreading mechanism. When we replicate our genome, there's a proofreading mechanism to avoid mistakes, but RNA viruses don't have that and they constantly mutate, which is not necessarily a problem. Coronaviruses are an exception because they also have a proofreading activity. So they actually mutate less than, for example, flu or mumps or measles. But the problem here is that we had explosive spread during the pandemic. And the more infections you have, the more replication cycles you have. And the more replication cycles you have, the more chance there is for a mutation. And then another thing that was found was that some people get infected, they're immunocompromised, so they don't make a good immune response, and the virus mutates faster in them, or not faster, but it stays around for a long time. It can become relatively persistent. This is very rare, but there are some cancer patients, for example, who show that. And this is shown here on the left side. So this virus was replicating in a person for 152 days. And if you take samples and sequence these viruses at different time points, you all of a sudden find these mutations that we also find in the variants. So this here is the ERIC mutation, which was found here, E484K, and the Nelly mutation here at position 501. And so the current thinking is that these variant viruses are actually coming from immunocompromised patients that have virus replicating in them for a very long time. And this is just to show you how these get detected. This is from South Africa. South Africa had a wonderful genomic surveillance from the start of the pandemic. They sequenced a lot of viruses. The US unfortunately didn't do that, but now we're starting to do that as well. And so in October, they found this yellow variant here, which is B1351. They just call it different here. And then you can see within basically 2 months that takes over the whole situation, right? So in November, already 90% of the virus that's circulating is this one variant. And so that's how we detect them. If there's explosive growth, we find them. And then they become a variant of concern. What do we know about the vaccines and the variants? So here in this table, I show you the variants on the left side, wild type or garden variety SARS-CoV-2. Then B117 from the UK, B1351 from South Africa, and P1. So for the J&J vaccine that we just started to use in the US, we know it's 72% efficacious against garden variety SARS-CoV-2. We don't know about B117, wasn't tested, but it's probably as good. And then for B1351, we know it's 64% efficacious. So there's a little drop, but it's actually not that drastic, which is great. And for P1, there's just no data. For the Novotex vaccine that should be licensed soon, the efficacy against wild type is 95%, drops a little against B117, but it's still 60% against this South African origin B1351. For AstraZeneca, it's different. AstraZeneca works well against B117 and garden variety SARS-CoV-2, but it doesn't seem to work against B1351 to prevent mild disease. And then for the mRNA vaccines, we have very high efficacy against garden variety SARS-CoV-2. We don't know yet against these variants, not just because nobody tried to look at that. The problem is that these clinical trials were done very early on when the variants were not around. But there's in vitro data, neutralization data, that suggests that those vaccines still protect against these variants. The important thing to realize here is against severe disease, all of these vaccines probably protect, even if you get infected with a variant. Now, how prevalent are these variants in the US? B117 became relatively prevalent, specifically in Michigan and in Florida. but it's still not that many cases. We have very little of it in New York. And again, these are not large case numbers, but it seems to increase in frequency. Again, this virus, if you're vaccinated, you don't have to worry about it. The vaccine covers it completely. P1, we see that there are very low case numbers in the US. It has been found 61 times, but it's not really looking like it's taking over. And B1351 has also been found 219 times, but again, it doesn't seem to be a big problem in the US right now. This might change over time, but right now it looks pretty good in terms of variants. And then, of course, we have our New York variant. It was a little bit disappointing because somebody from California identified this variant, and as a New Yorker and researcher working in New York, that kind of hurts. We wanted to identify it ourselves, but okay. And so what happened with that variant? It's called B1526, and it comes in 3 different flavors. And these are calendar weeks here on the x-axis. It started to increase in frequency, and that's why people started to look at it. But then it kind of levels off now. So it doesn't seem to have a big advantage, and it doesn't seem like it's exponentially spreading right now. And we also don't think that this is going to be a problem for vaccines at all. Just a few numbers to finish. Right now, approximately 485 million doses of vaccine have been used globally. Most of them are RNA vaccines. Israel has covered 54% of their population. That's huge. They're really kind of doing an awesome job. With that, there are some smaller countries like the Seychelles and the Maldives, for example, that also are already in that percentage range, but they have, of course, a much smaller population. And in the US, we are doing pretty well as well. We're now to 20% of the population covered, and it's increasing fast. And I think that will really help to make the situation much more normal in the coming weeks and months. Just one piece of information before I end here. These efficacy data that we get from the clinical trials are, under, you could say, ideal conditions, right? You have a clinical trial, a lot of things are defined. Usually you have relatively healthy people in these clinical trials. And so we got relatively high efficacy, 95% and so on and so forth. But then when you start to use a vaccine in the population, you can also measure how well it works. And we call that effectiveness. So there's efficacy from a clinical trial and effectiveness in the population. And effectiveness is usually a little bit lower than efficacy. But now we're starting to get data mostly from Israel, but also from Scotland, that suggests that the effectiveness is almost as high as the efficacy. So here's an example for the Moderna vaccine, for the Pfizer vaccine from Israel, where they measured 94% effectiveness in the population. And that is really, really nice to see. And it suggests that these vaccines work really well, also in real world scenarios in our population. So with that, I'm going to stop. Just in summary, vaccines against SARS-CoV-2 work and protect well against disease. There's data from 10,000s of people in phase 3 trials and millions of vaccinated people that suggest that these vaccines are very safe, very comparable to other vaccines that we are using for many years now. And hopefully these vaccines will bring us back to normal life in the next few months. I think there are some countries like Israel, but also the US and the UK that are really on a very good track to get there. Unfortunately, there's a lot of countries that right now don't have access to a lot of vaccines. And I think we also need to change that, and we need to make sure that we can get vaccines globally distributed, because if we are safe here, but there's a lot of virus replication somewhere else, and there's a variant that all of a sudden arises and then comes back and the vaccine doesn't work against that anymore, we have to avoid that. We have to make sure that everybody's covered in all countries. And my last point is, if you have a chance, and I know you're teachers, so you do have a chance, get vaccinated. It's worth it. And with that, I'm happy to start answering questions.
All right, awesome. Thank you so much, Dr. Krammer. That was fantastic and super informative. I'm going to hand it off to my colleague, Uzma Shah, our program officer for professional development, who's going to help run our question and answer section now. So I'll hand it over to Uzma.
Thank you, Jen, and thank you, Dr. Krammer. We're so pleased to have you back tonight and sharing all of these updates and questions around safety and the variants. And we have a lot of questions that folks have already pasted in the chat, but we'd love to start with anyone who'd like to directly ask a question of Dr. Krammer. If you would like to ask a question verbally, just either use the hand raise feature. That's the easiest way for us to find you. And then I will signal you and you can unmute yourself and ask your question. So let's begin with anyone who would like to directly ask a question. I see Hanny. Hanny, go ahead.
Hi, good evening, doctor. My question is a tangent, but early on, especially when the virus was really soaring in Europe, they released papers indicating that for some reason, people who smoked cigarettes, they saw a much lower rate of hospitalization. And they looked at papers from France, China, and Israel regarding this. Was there any follow-up to any of this? Did they ever figure out why that was the case? Or have you heard anything about this?
Yeah, no, you're right. There were a couple of studies initially, also from relatively large ones from China, that suggested it was statistically significant, but the effect was not really big. That smokers were a little bit protected from severe disease, but there is no other studies that suggest that that's not the case. So you always have to be a little bit careful with if the effect size is small, right? And it was small in these studies. It could just be a bias, and it turned out it wasn't really the case. So that hasn't been confirmed.
Okay, thank you.
Thanks, Hanny. Let's go to Susan. Susan, go ahead and ask your question.
Hi, I was wondering, so the initial data for pregnant women with these vaccines looks promising, but is there any additional information you have about that, women who are pregnant getting the vaccine?
So there's no additional information right now. We knew, usually if you're part of a phase 3 study, you sign a piece of paper saying that you take contraception, you're not going to get pregnant. But of course, always people get pregnant, right? And so there are cases, a handful of pregnancies in the Moderna trial and the Austro-Seneca trials, in the Pfizer trials. And you can, of course, compare that to people in the placebo groups. There was really no difference in any of the outcomes. But I think Pfizer is actually running a trial to evaluate that further. But right now, the CDC recommends that, if you are exposed to the virus, if there is a risk that you get infected, you should discuss that with your physician if you want to get vaccinated. They didn't say pregnant women should get vaccinated. They said they should discuss that and then make a decision. But there's no biological reason to think that there should be an issue. So, I mean, the studies are ongoing, but right now or by now, a lot of pregnant women have been vaccinated, and the other point is that you might actually protect your baby too, because there's antibody transferred. So yeah, there's not much more to say about it. Right now, there is no reason to think that there's an issue.
Thank you.
Thanks, Susan. Let's go to Donna and then Anne. Donna, go ahead.
Okay. Hi, Dr. Krammer, and thank you so much for sharing your time with us. This is just a great seminar. I'm wondering how much we know or don't know about the efficacy of the vaccine in people who are immunocompromised, the people who are generating all the variants. If we vaccinate them, will they mount an immune response, or will they just keep generating more variants?
Yeah, that's an excellent question. The problem is that immunocompromised is not the same as immunocompromised, right? There are people who are immunocompromised a little, but make pretty appropriate immune responses and would be completely protected. And there's other people who have basically issues with their immune system, and they don't mount an immune response at all. And then you have the whole spectrum between, right? And so there's right now really not that much data about who makes a good immune response, who doesn't. There are studies ongoing. So some of these vaccine companies selected specific groups that they want to look at, and there's going to be data for those specific groups. But again, it's going to be hard to generalize that. And then the issue is also that some people get biologics that suppress the immune system, but they don't get it all the time. And then it might also depend on the timing. I think the best way to prevent that people with a compromised immune system get infected is to make sure that there's little virus circulating in the population, which we, can manage by social distancing, wearing masks, and for all the healthy people, getting vaccinated.
Great.
Okay, thank you.
We'll go to Anne and then Keith. Anne, go ahead.
Okay. Thanks for this talk. It's been great. My question was about the structure of the variants. I was particularly struck by you pointing out that several of the variants seem to have like mutations in like the same place in the structure. And I was wondering if there was anything about the structure of the virus that like made it more likely that certain mutations would occur.
Yeah, so what you're pointing out is there's convergent evolution. And there's already data that says that if you get infected with the B135 from South Africa, you make a good immune response, of course, against that virus, but also against P1 from Brazil, because the mutations are very similar. So again, we really don't understand this yet. It seems that all the same mutations are coming up in all these variants of interest or variants of concern. And this might just be further adaptation to humans. That might be the case. There are some people who actually think that there is immune selection also involved, that people make some kind of weak immune response and then select for viruses that can overcome that. But it's not really clear. There's two scenarios that we could go into in the future, right? One would be that, the viruses go in one direction and then stay there just because they like these mutations, and then they stay stable there. And that's what I would expect. And then the other possibility is that they keep mutating. And that's not what we typically see with coronaviruses, but, this virus surprised us already once, so I wouldn't bet on it. We'll see.
Great. Thanks, Anne. Keith and then Rich.
What type of cells actually pick up the RNA vaccine, and how is it specific to certain cells, or is there a receptor that the RNA vaccine goes to? Does it go to
Yeah, there's super nice mouse data for that. Of course, with the other RNA vaccines, everybody was trying to figure that out already. And with mice, you can do all of these things. We can't just dissect the muscle of somebody who was vaccinated, but with a mouse, we do that. So basically, most of the RNA is taken up at the injection site, right? It's mostly myocytes, so muscle cells that take it up. And then there are some antigen-presenting cells that take it up too. And those we think are the important ones. The muscle cells will make the protein, but they might not contribute a lot to the immune response. The antigen-presenting cells that take it up then basically migrate to the lymph nodes, right? And so usually they are training lymph nodes that you get from an infection. They're basically in that area here, right? And so those cells bring these antigens there and basically start to educate B and T cells in those lymph nodes. And those are probably the cells that are important for making a good immune response. We see that these lymph nodes, usually if you make an antibody response or a T cell response, it happens in lymph nodes, right? And we see that that works very well for the RNA vaccines. Much better than for flu vaccines, for example.
Thank you.
Great question, Keith. A lot of folks had that same question, so thanks for asking. Rich, and then we'll go to Pete.
I was wondering, what are the current recommendations regarding using antibody titer testing to determine if booster shots are needed in the near future?
Okay, a super good question. we're working on that. The CDC is working on recommendations for that. The issue in the beginning was a little bit that everybody was using different tests, and we need to establish something that's called a correlate of protection, right? We need to figure out what the protective titer is. But because everybody had different measurements, we couldn't do that. But the WHO usually develops an international standard for that. And now all the tests that are used are actually getting calibrated to that standard. And then you get the same units from all the tests. And that will make it easier to figure that out. But what you're talking about is something that has been implemented for a lot of different other vaccines, right? For measles, for example, you can get tested and somebody can then tell you, oh, you need a booster shot, or you're still good and you're protected. But that will come. And with that, I expect that there's also going to come a lot of changes in terms of policy, because then you can actually tell a person if the person is likely protected or not. I think we need to wait a few more weeks, but the CDC is working on recommendations.
Thanks, Rich. We'll go to Pete and then Tamana.
Thanks, Uzma. So you saying that the mRNA vaccines and all the data shows that they're so efficacious and so effective, and the technology has been around for so long, or not that long, I guess, in the medical world, but do you see more companies going down this route with mRNA vaccines for other types of viruses? And if so, with the speed of this vaccine coming out, would that apply to other vaccines for other viruses?
Yeah, that's an excellent question. And the answer is yes and no. So, I mean, it's amazing to see how quickly this could be done, right? What is not going to happen is that vaccines that we already have are going to be replaced by that, just simply because the vaccines we have work. If you look at MMR vaccines or pertussis or whatever, they work very well, and developing a new vaccine costs a lot of money, right? So if you would develop now a measles vaccine based on mRNA, that would cost you probably $500 million, right? So there's no need to do that. In terms of other vaccines for things that are challenged right now, I'm talking about HIV, tuberculosis, also CMV, rabies, malaria. I wouldn't see it as a game changer, but it could contribute to speeding up to get to those vaccines, right? It's not going to solve all the issues, but it might be a good tool that helps us to move forward. For pandemics, I think it's going to be the standard tool for the foreseeable future, just because you can do it so quickly. And I think and the problem is there will be another pandemic. I hate to tell you that, but this is not the last time this is going to happen. But the next time it could probably be done in 3 months. And that would probably, save a lot of lives. So for that, it's really good technology. There's another issue. It needs to be further optimized, right? Because there's two disadvantages. One is the storage needs to be frozen, and other vaccines don't need to be frozen. You just keep them in the fridge. And that's really a big issue. Not in the US necessarily, but globally, right? And the other issue is this reactogenicity. I don't know how many of you got the mRNA vaccines, but many people, specifically after the second shot, don't feel that well. And, I would prefer to get vaccinated with vaccines that, don't make me feel sick for even if it's just for a day and I know that it's not dangerous, right? So it might need to be further optimized so that it can be stored at, 2 to 8 degrees Celsius in the fridge, and so that it doesn't do that, right? Because nobody wants to feel sick after getting vaccinated.
Okay, thank you. I think we may have lost Uzma, unfortunately, to a Wi-Fi mishap, so I'm just going to step in here.
I was sorry.
Hi. Look at Tamana's up next. I'll put you on the screen.
Okay, sorry. Hi, thank you again. I was thinking about just how the mRNA vaccine was encapsulated in like the lipid nanoparticles. So like assuming it's protected against like nucleases, like degradation. Do you know of like other forms of encapsulation that they're like working with, or is this like the only type we have so far? It's obviously very efficient.
There are many different ways of doing that. The first RNA vaccines that were used were actually complexed with protamine, so they were not in lipid nanoparticles. There's different ways. There's actually nice reviews out there that look at that, and of course, a lot of them have been tested in animals, right? So some ways to formulate it work better, some work worse. And so there are actually a lot of options to change that. There's also a way to complex them with the RNA with peptides. Actually, my lab is trying to do that right now, because then you could give that RNA as a nasal spray. So there's many ways, and it can be optimized in the future.
Okay, great. I'm going to go over to Adam Chawansky next.
I was wondering, what do you see as the long-term future of COVID? Like, will we eradicate it via global herd immunity, or will it converge on like a single variant that includes like all the different mutations? Or is it just going to be like the flu, where there's like a few strains that we try to vaccinate against every like cold, flu, and COVID-19 season?
Yeah, so we've only eradicated two viruses so far. One was smallpox, and the other one was rinderpest virus. That was a cattle virus. You can't eradicate viruses anymore. I'm just going to say it like it is. When smallpox was eradicated, people were not asked if they want to get vaccinated. They were vaccinated. You cannot do that anymore. And I don't think anybody should be forced to get vaccinated. And so there will always be a proportion of people who is not protected just because they don't get vaccinated. So what I see for the future for SARS-CoV-2 is the same that we have with the other human coronaviruses. It will just become human coronavirus number 5. Once everybody has some immunity, and even if there are variants that might reinfect people, it's not going to be a severe disease anymore. And it will, over time, not because the virus attenuates necessarily, but because we build up immunity, it will become in my mind, it will become something that's relatively harmless, but might, be the annoying 5th coronavirus that causes common colds. And the other point that is important is, you see that a lot of kids get infected and they have mild or asymptomatic infections, right? But they build up immunity too, and they might get reinfected when they're 20, and then maybe again when they're 40, and maybe again when they're 60. By the time they're 80, they have so much pre-existing immunity that this is not going to be a severe disease anymore. And so that's probably what happened with the other human coronaviruses, which when they were introduced into the population, likely also caused the pandemic, right? One of them, OC43, probably caused the Russian flu of 1890, which killed a lot of people and then just became a common cold.
Okay, great. I'm going to go over to Jesse John. You can ask your question.
Hi, how are you? Thanks for the beautiful talk. My question is pertaining to one, will you be able to share the resources that you presented tonight? Because I would like to look at some of those papers. And then 2, how do they go about calculating the length of immunity that we will have from these immunizations? Because we know with SARS-CoV-1 that T-cell immunity lasts for a couple of years. And I'm just wondering what that process looks like for them to calculate the length of immunity for SARS-CoV-2.
So in terms of the resources, I don't have a reference list or something like that. There is a paper out there that gives a nice overview over the vaccines that were in development. It's a review that should be freely available in Nature. And I think for most of these things, I had some kind of reference. So I think that could be made available. But in terms of the calculating, we can calculate that. You need to follow it, right? And so it's a real-time assessment. You can calculate it once you know how it develops over time, right? So for hepatitis, for example, we know how it develops over time in many people, and then you can start to model it, and then you can measure your titer, and you can say, now I have that titer, and this is going to last for, 36 years. But you need the primary data first. And so we need to do that in real time. There's not going to be any way to calculate that. People will have antibody measurements as part of their phase 3 trials now for at least 2 years. So I'm in the Pfizer trial. I kind of enrolled in that when they started, and they will follow me for at least 2 years, right? So I still have to come in and deplete me and whatever. And you can also do that on a population basis. So we'll generate that data as we go. But it's complicated because people might get reinfected asymptomatically, and that boosts their immune response again, right? So it's actually not that easy to just calculate it.
Is there any way that we can draw correlations from the fact that SARS-CoV-1 utilized the ACE2 receptor and SARS-CoV-2 binds tighter? Is there any way that we can sort of model that, or is that just they're too different and we need to only rely on real-time models?
No, and you're referring to papers that looked at a very small number of people, right? 8,000 people were infected globally with that virus. The studies that have been done were, in a handful of people, and you can't really extrapolate from that. It's nice to see that there's another paper that I know. There's one paper that says, oh, T cells are still there. There's no antibodies. There's another paper that says, oh, after almost 20 years, there are still antibodies, right? So that data is not solid enough to model anything based on that.
Thank you for your time. And is it possible for you to share that PowerPoint with us? Because a lot of us are teaching pandemics currently, and it would be good to model it using some of the graphics you have. If not, we completely understand.
Yeah, no, the slides I can share, no problem. I don't know if MFA would kind of be the resource where I deposit that, but that's not an issue at all.
Yeah, if you share that with us, we can make sure to make that shareable for everyone in attendance.
I appreciate your time. Thank you.
Thanks, Jesse. Okay, looks like we have a couple more questions of the hand raised, but I do want to do one from the from our chat before. Looks like Astrid's son had a question about masks. Astrid, I don't know if you want your son to ask on camera, but I can also ask it if you'd like. Okay, I'll go ahead and ask it. They were wondering, what is the difference based on data in areas where masks are being worn versus areas where masks are not really being worn or are being worn less frequently?
It's hard to say. There are not really good studies on that. What you can say is that areas where masks are worn on a regular basis, including Japan and South Korea, have very few problems with the virus. So you can generalize from that. I haven't seen any. No, there are probably some, but I haven't read them. There are probably some studies that looked across a lot of studies that looked at it, but I don't remember what the data looked like. I think they suggest that it works, but yeah, it depends on the setting. It depends on how much virus is around, which masks are worn, who wears them. As medical personnel and researchers, we have a really good track record of not getting infected if we wear N95 masks. But if you wear a regular mask over your chin in the subway, it's not going to protect. So the masks work. I would need to go and dig out papers to answer that.
Okay, great. I'm going to hand it over to Jesse Markowitz for your question, Jesse.
Hi, thank you very much. This is super informative and helpful. I wanted to know if you have any suggestions or if there's much research or resources currently on how to address vaccine skepticism or people who are very reluctant, if that's something that's, or I guess what are more effective versus less effective ways of having those conversations, because it's conversations that I am eager to have, but understand that just throwing a bunch of information at people is not likely to convince them.
Yeah, it's hard. First of all, there are people who actually just research that and try to figure out how to do that best. Dan Salmon, for example, at Johns Hopkins is one of those people. That's the research that they do. I don't know. I mean, my approach is always to talk to people and make sure that they get all the information they need, and then they can make their decisions by themselves. But you know, I think there's skepticism and then there's anti-vaxxers, right? And those are also different things. With people who are skeptical about vaccines, usually I know a few of them, and some of them now got vaccinated. Usually it helps to really discuss the data with them, to talk also about the risks that COVID brings with it, which, are substantial. And I think, if people get enough information, they make the decision to get vaccinated. Anti-vaxxers are a different story, and I think it's not even, from my perspective, it doesn't even help to talk to them. So again, this is more going into social sciences. I have the same problem, but I think the more information that can be shared, the better, and specifically if that information comes also from independent sources and not just from the vaccine companies or the government.
Thanks.
I've said bad things about vaccine companies in the past, and they will do it if they annoy me. And for example, AstraZeneca right now is this basically back and forth and showing the wrong data. I criticize them a lot, right? But I think, independent sources are good.
Thanks, Jesse. So we have one question that was being talked about in the chat a bit that comes from Fung. The CDC recently changed the recommendation for social distancing in schools from 6 feet to 3 feet. Do we have more information about COVID infection in children and young adults? In addition, what is the current progress in the vaccine clinical trials for this group?
There's a lot of progress in the clinical trials. Moderna went down, and they're now in really small children. They're going down to 6 months of age to assess the safety of the vaccine. They don't need to look at efficacy. It's really about safety, right? Pfizer announced today that they're going to start in the same age group. So we'll have probably vaccines by the end of the year, but probably not that much earlier. Maybe September. It's not really clear. It's also not clear for which age groups these vaccines then will be safe, right? It could be that it's just safe for 14 plus. So we'll see. We'll need to look into that. The problem is a little bit that reactogenicity, which is already a problem with these vaccines, is usually much more pronounced in kids than in adults, and old people have the least of that reactogenicity. So there might be an issue. About the 3 and 6 feet guidelines, I'm not 100% sure why they were changed and what the underlying data is. What we do see now is that there are a lot of infections in kids, and there's a lot of transmission in kids in Europe. It's unclear if that is the B117 variant, which is a little bit more infectious, but there are problems, and there was a very heated debate in Europe about closing schools in many countries. This was really the last thing they wanted to do. But now they are starting to close schools in some areas because there are transmissions, and there are clusters in kids. So I can't really answer this question well, but not sure what the information was that the CDC has in order to change the regulations.
Okay, great. Thank you. Ava, I saw your hand raised, or you're all good?
That was my question, but I think in general, I've been seeing in the chat, I had similar questions that were coming up in the chat about schools and kids, like that, another one that Danielle just put in there, just about, all of us are teachers and we're thinking about, reopening, and obviously with vaccines, we're safer doing that. I just think I've been having a lot of conversations with folks who have kids, or want to get their kids back in school, or are nervous to get their kids back in school. And there's some studies that are saying kids generally don't get infected, and then other studies that are saying, oh, look at all these kids that did get infected. Danielle's question is also a good one about ventilation in schools and the importance of that. So I guess, could you speak a little bit more to, what you've been seeing about schools opening? You kind of already did, so I don't want to.
Kids do get infected. The big question still is, how much do they transmit, right? And that depends on the age group. Older kids, 14 above, there's no question, they do transmit like adults. Younger kids, it's still not clear. If you see clusters, and as I said, there was a lot of that in Europe recently, it's sometimes not clear if a teacher infected the kids or if the virus was spreading among the kids. There are cases where kids infect adults that are proven, and even like small kids, even toddlers have infected adults. So we still need more data. And I think the situation in New York right now looks really good. There's actually very little infection in kids in schools, at least to my knowledge. And it's not clear to me if this is really because it's better managed or because the variants are not really, expanding that much here, or, there might be all kinds of differences. In terms of ventilation, that's an important point. The higher the ventilation rates, the quicker you get rid of aerosols. And I mean, that's one of the reasons why everybody was saying outdoors is much safer than indoors, right? Outdoors, you have so much air movement that, the chances that you get infected are very, very small. Indoors, it's bigger, but the more ventilation you have, the lower the chances.
Great. Thanks, Ava. Okay, it looks like we have time for maybe 2 more questions. And I'm going to go to our chat for that. We have a question from Suzanne and Jessica. They ask, how often does a positive COVID test get tested to see if they are variants? Is a PCR test able to determine which variant you are infected with?
So yes and no, it depends on the PCR test. So the reason why the UK was able to quickly determine how fast that variant was spreading there was because their PCR test that they were using had 3 targets. So you get 3 signals from that test. And the virus had a deletion. And that means that you only get 2 signals. And that makes it very easy to track. Typically in the US, what we do to find out if it's a variant, you can do that via PCR. But the standard testing will not tell you if there's a variant. Usually these viruses then get sequenced. And for example, Mount Sinai is now trying to, or actually successfully doing that for some time. We just randomly sequence 10% of the viruses that we get. That gives you a pretty good idea. But again, it's not typical that you would get a diagnostic test and they tell you, oh, it was a variant or not. That is done in some areas in Europe where there are variants that are coming up, where they really want to track that right away, but not necessarily in New York.
Okay. We have one from Karen. This is also being talked about a bit in the chat. She had been getting a lot of questions about if a vaccinated person is likely to carry the virus, but not have symptoms. And so she wants to know how she should go about answering that question.
Yeah, so the chances are lower that somebody who is vaccinated gets infected. But it might still happen. If it happens, that person is very likely carrying less virus and for a much shorter period of time. And that translates into a relatively small chance of basically giving that virus to somebody else. And that's also why the new CDC regulations or guidelines say, it's not a problem if 2 vaccinated people meet without masks and have a nice dinner, right? Not a problem because they're both protected. They might actually spread the virus among themselves, but there are no consequences, right? What you have to keep in mind is when we talk about a 95% efficacy, that means that out of 100 people, 5 still got infected, right? So what these vaccines do is they reduce your risk. And even for symptomatic infection, it's just a risk reduction. So it doesn't remove all risk. So even if you're vaccinated, you might still get sick. It's just very unlikely. And the same is true with the asymptomatic infections.
Okay, and I think I do have one question from Uzma from her poor Wi-Fi. She wants to say, while many Western nations have ample access to COVID vaccines, what's the prognosis for folks in developing countries? And how are pharma companies addressing the issues of access and equity?
Yeah, it's a problem. When the vaccines were developed, COVAX was formed, which is basically an initiative that makes sure that all the vaccines are going to be shared throughout the globe until everybody in each country, until the vaccination rates in each country are at 20%. Countries could participate in COVAX. That was also sponsored by the Gates Foundation, CEPI, and so on and so forth. And some companies basically took part in COVAX, some didn't. And now it turns out that this doesn't work that well. the ones who have the money grab the vaccines. Vaccines are mostly now delivered to the US, Israel, and the UK. Even the European Union has issues to get their vaccines. But nevertheless, there are shipments that are going out to other countries. And what people have to keep in mind when we think about pharma companies, we think about US and European companies. And to a certain degree, that is true. Pfizer is a US company, Moderna is, Austro-Seneca is British and Swedish, and so on and so forth. But the biggest vaccine producers actually sit in India and in China. And they are actually delivering most of the vaccines currently to low and middle income countries. And Serum Institute in India, for example, is making the AstraZeneca vaccine, right? So they have the same technology and they are providing it globally. And they're part of that COVAX consortium. But I think we need to do a better job with that. It's really terrible to see what happens there, but the truth is that the countries that paid most for the vaccines are getting most of them, and they're getting the most doses. I think in the future, what we need to do is we need to make sure that a lot of countries actually have capacity to make their own vaccines. I think that's really the way to go forward, because if you have a country and the country can produce vaccines for themselves, or at least for a region, that works out well. But yeah, it's not an ideal situation, and I have no clue how to solve that politically. I'm a virologist.
No, that's a very thoughtful answer. Okay, I think that's it for us. Thank you so much, Dr. Krammer. This was fantastic. I think this was so informative, and clearly from all these questions, our teachers got a lot out of this. And I think the takeaway is we should go get vaccinated when we're eligible. They're safe, and they're going to really do a lot of good. So thank you so much for taking time to talk with us tonight.
Thank you.